studying in K.T. Patel College of Pharmacy. In this lecture, we are going to talk about bioassay. Let's start it. First of all, we are going to completely point in this video. First, introduction, principle of bioassay, application of bioassay, types of bioassay, bioassay of insulin, and last point is rabbit model. So let's start with some basic introduction. What is meaning of bioassay? Bio means living and assay means to test. Definition: bioassay is defined as the estimation of the concentration or potency of a compound by measuring its biological response to determining the physiological effect of substances on tissues, organ, or living organism. Bioassay can be used to measure concentration of a drug as its potential relative to another drug. Bioassay may be performed in two parts. First one is in vivo and second one is in vitro, having an important role at all stages of drug research and development, and are used to estimate the potency of a drug as well as toxicity. Bioassay can be used to measure the concentration of a drug as its potential relative to another drug. Now looking at principle of bioassay. The basic principle of bioassay is to compare the test substances with the international standard preparation of the same and to find out how much test substances is required to produce the same biological effect as produced by the standard. Active principle should show the same major response in all animal species. Pharmacological response produced should be reproducible under identical conditions. Example: adrenaline shows same rise in blood pressure in the same species under identical conditions: weight, age, sex, strain, and breed, etc. The reference standard must own its activity to the principle for which the sample is being bioassayed. Activity assayed should be the activity interest. Individual variation must be minimized. Bioassay might measure a different of the same substances compared to chemical assay. Example: testosterone metabolites. Next point is application of bioassay. The tissue is determined by plotting concentration response curve with unknown agonist, which helps not only to determine the concentration but also the potency of the sample. It is especially used to standardize drug, vaccines, toxins or poisons, disinfectants, antiseptic, etc. This also helps to determine the specificity of a compound to be used. The concept and principles of bioassay can be very useful when performing structure activity relationship studies as part of the drug discovery process, especially when a relative potency and functional effect of different compounds on intact tissue system are to be tested. Testing of infected patient spectrum helps to determine which antibiotic to be given for quick recovery. It is simple and faster method. Amount of test drug required is small, does not involve complicated calculations. Certain complex compounds like vitamin B12, which cannot be analyzed by simple assay techniques, can be effectively estimated by bioassay. Sometimes the chemical composition of samples is different but has some biological activities, and for sample where no other methods of assay are available. Then next point is type of bioassay. There are two types of bioassay. First one is quantal assay and second one is graded assay. Quantal assay. A quantal assay involves an all or none response. For example, insulin-induced hypoglycemic or the digitalis-induced cardiac arrest. Digitalis-induced head drop in rabbits. The drug producing quantal effect can be assayed by endpoint method. The response is either positive or negative, as there is no intermediate response. The quantal method is commonly employed for compression of effective dose or median lethal dose. Second part is graded assay. Graded assay are based on the facts that increases in the concentration or dose of a particular drug. Proportionality increases the observed response. The drugs producing graded response can be assayed by two types. matching or bracketing method, graphical method. This graded assay is based on the nature of the effect, the substances that is expected to produce. For example, contraction of smooth muscle, preparation for assigning histamine, or the study of blood pressure response in case of adrenaline. The choice of graded assay depends on three parts. First one is precision of assay required. Second, quantity of the sample available. Third, availability of the experimental animals. Now, looking at bioassay of insulin. Insulin means it is a pancreatic hormone which released for pancreatic beta cell and help to control the blood sugar level by converting glucose into glycogen. It was discovered in 1921, which helped millions of people suffering from type 1 diabetic mellitus. In IDDM means insulin dependent diabetes mellitus, capability of insulin production by the lost. Next point is standard solution. Standard solution is prepared by using pure dry and crystalline insulin and unit contains 0.04082 mg. This unit is specified by Ministry of Health, Government of India and is equivalent to international unit. Second point is preparation of standard solution for bioassay. 20 unit pure insulin is dissolved in small volume normal saline and acidify it with HCl means hydrochloride to pH 2.5. 0.5% phenol is added as preservative. Add 1.4% to 1.8% glycerin. Final volume should contain 20 units per ml within 6 months. Then last point is rabbit model. Animal selection. Experimental protocols should be approved to the experiment by IEC, Institution Animal Ethics Committee. Thereafter, healthy rabbit 2-3 kg are selected and habituated for the experiment in standard environment condition as per the norms of CPCSEA means Committee for the Purpose of Control and Supervision of Experiment of Animals, Government of India. After that, animals are kept in fasting for 80 hours before performing the assay. Water is withdrawn during the experiment. Next point, standard and test sample dilution. 1 unit per ml, 2 unit per ml is freshly prepared for stock solution of standard and test solution. Next principle, the potency of a test sample is estimated by comparing the hypoglycemic effect of the sample with that of the standard preparation of insulin. Next, first part, the test. First one is A. Initial blood sugar level. A sample of blood is withdrawn from marginal air vein of each rabbit and examine the reducing blood sugar level before insulin treatment. Second, B. Final blood sugar level. After standard and test insulin injection from each rabbit, a sample of blood is withdrawn up to 5 hours. At the interval of 1 hour, each blood sugar is determined again. This is known as final blood sugar level. Second part of the test, crossover test. The same animals are used for the second part after 1 week. Again, animals are fasted and initial blood sugar is determined. The grouping is mean. Those animals which received the standard are given the test and those which received the test are now given the standard. Those animals which received the less dose of the standard are given the higher dose of the test samples in vice versa. This test is known as twin crossover.